All right, hi everybody. Today we're going to talk about the cell, and so we're going to talk about the cell structure and function. So today we're going to get talk about the cell basics. So we're all made up of cells, right? Like for example, like we have our brain cells that are firing and help us think and understand what other people are saying. We have muscle cells that help us stay upright. We have heart cells that are pumping blood through our body. And so today we're gonna basically talk about not only what cells are made out of, but how have they evolved, how are they organized, and why the organization is important for life. All right. And so like I mentioned, mostly everything we do requires cells. And when you think of an organism, so a living thing as a whole, then we need to think about what our cell does in the micro level, right? So cells reproduce, they grow and develop, they need energy, they respond to the environment, and they require homeostasis, which is an internal balance. All right. So we need to reproduce and make more of ourselves. We need to grow and develop. We need energy to power all of our activities. We need to respond to our environment. Maybe move food towards our mouth or move when we sense danger, right? And like I mentioned, homeostasis is just that internal balance. So even though your external environment changes, your internal conditions stay relatively stable. All right. And so the cell theory, I'll talk a little bit about the cell theory, which is one of the most important principles. So this is a contribution due to many scientists is why we have the cell theory so Schleiden, Schwann, and Verschoor. And so in the 1800s, these scientists, they started looking through the little microscopes and they started making observations, right? So Matthias Schwilden was studying plants and he noticed that they were made up of little compartments. And Schwann also found the same thing in animals. So then that's when we knew that plants and animals have cells. Um, Verschoor over here, he made an important realization where he kind of noticed where cells don't just disappear or appear, and so they come from other cells. And so that's when we came up with the cell theory. All living organisms are made up of one or more cells, and cells come from pre-existing cells. So basically, you could technically trace your lineage all the way back to the first cells that appeared on Earth billions of years ago. So, yeah, all living organisms are made up of one or more cells. All cells arise from pre-existing cells. And so, like I mentioned, you need a microscope to see cells. You can't just see your cells with your eyes, right? And so, cells are super, super tiny. They're measured in micrometers. Um, and so, to put it in perspective, if a typical human cell were the size of a basketball, then a human being would be about 15 miles tall. So cells are super, super tiny, and that's why we need microscope, sorry. And so here you can see the range of different tools that you can see. So your naked eye stops around, I want to say, 100 micrometers. And so light microscopes, like the ones we use in class, right? They have lights. It could get us down to seeing bacteria and even some organelles. But for like really, really, really detailed things, we need an electron microscope. And so here are the differences between like a light microscope, the one we have in class, and an electron microscope. The thing with the electron microscope is that it kills the cells, so you can't watch living processes. So we have to use different tools for different experiments. Um, yeah, and so here you can notice how with a light micrograph you can see the cell wall, parts of the cells inside. But when you get down to like a TEM, which is a transmission electron microscope, and a CEM, which is a scanning electron microscopy, then you can see that you notice more details. And honestly, it looks super, super cool. Like you can see the 3D surfaces. But like I said, with the electron microscope, it kills living things. So there's a trade-off. All right, and here you can see cells. All right, and then just to go over the objective, today we're going to discuss the structure and or function of cellular components and organelles. We're going to describe similarities and differences between prokaryotic and eukaryotic cells. We're going to describe the relationship between the functions of endosymbiotic organelles and their free living ancestral counterparts. So we're going to talk about the ancestry and where cells came from. And so I want you to think to yourself, why do you think different types of cells have very different structures in organisms? And so think about the question and share what you're thinking with someone near you. And so what is a cell? A cell is a group of organelles and molecules working together and perform a specific task and help the organism maintain homeostasis, right? A balance. And so here you can see a couple of organelles and some molecules. And living organisms must at least have one cell. And there's two categories, prokaryotes and eukaryotes. So the root word pro means before, and karyo means nucleus. And so that's before the nucleus of the cell. So things like bacteria, archaea, those are prokaryotic. They're usually smaller, which we're going to talk about in a little bit, and have genetic material, so floating around in the cytoplasm. And then eukaryotes, U means true, and it has a true nucleus where the DNA is found inside of a membrane. So like you could think of things like plants, animals, fungi, protists. So the way I remember is you have eukaryotic cells. All right. So yeah, so let's talk about some similarities and differences between prokaryotes and eukaryotes. So like I mentioned, bacteria and archaea, it's single cell, no nucleus, no membrane-bound organelles, smaller and simpler, and they're the oldest cells. Eukaryotic cells include plants, animals, fungi, protists, as I mentioned, are multicellular, so they have a lot of cells. It has a nucleus. It has membrane-bound organelles. Things aren't just free-floating around the cell. And they're larger and more complex cells. All right. And so we know that eukaryotic cells evolved from prokaryotic cells. So this is like before the nucleus, and this is like after. Once we have a nucleus, because we have eukaryotic cells, we have a nucleus. And so the things I want you guys to remember here is that both prokaryotic and eukaryotic cells have genetic material, so DNA, RNA. They have cell membranes. They have ribosomes. And then some prokaryotic and eukaryotics have cell walls, flagella. They both use cellular respiration to make ATP. All right. And here is the same chart, just with more visuals, so you can see, like when I mentioned that they both have cell membranes. This is typically what a cell membrane looks. And then we're going to talk about all of these little parts throughout the unit. So you are going to have to memorize each of these structures and their function. So like we mentioned, cells are super, super tiny. It can range from 10 micrometers to 1 millimeter in range. And we're going to talk about why they have to be so tiny. But cells have to be big enough to fit all the DNA organelles, but they also have to be small enough to efficiently exchange nutrients, oxygen, and waste with extracellular environment to maintain homeostasis. So extracellular just means outside of the cell. So it has to exchange things throughout. Really quickly, just for a quick analogy so you understand why cells have to be so tiny, you could think of like a big house. It takes a lot of, for example, if you want to turn on the AC, it takes a lot of time and energy for the whole house to get cool. And so having it be smaller makes it more efficient. And we're going to talk about this when we do a surface area to volume lab. All right. All right, let's get into cell shape and function. So a cell shape and the amount of types of organelles it has determines the function of the cell. So basically, like for example, you can have a sperm cell that's going to have long flagella and a mitochondria because they need energy to swim towards the egg, right? Plant cells are often rectangular and they stack together to provide structural support. So the shape and everything it has inside, so its internal organization, directly relates to its job and the function of the cell. All right. And so this brings us to compartmentalization. So eukaryotic cells have internal membrane that compartmentalize their function. So what this means is that basically, like in a house, right, you have different rooms. Like your bedroom is for you to sleep, your kitchen is for you to cook things. And so basically the cell has different rooms that specialize and have specific jobs. And so basically eukaryotic cells are like this. So we can classify organelles into two categories. You can have something that is membrane bound and non-membrane bound, all right? So non-membrane bound organelles include ribosomes and cytoskeletons in the cytoplasm. And so what non-membrane bound organelles means, it means that they don't have their own membrane inside of the cell. Like I mentioned, ribosome, cell membrane, cell wall, cytoplasm. And a membrane-bound organelle would be an organelle that has their own membrane inside of the cell. So a nucleus, a vacuole, a mitochondria, a chloroplast, a Golgi, and ER. And so this is because you can create different chemical environments in different parts of the cell. So for example, inside of a chloroplast, you can have a different pH and ion concentration than the cytoplasm, right? And so it allows it to react differently. And like we mentioned, things that have membrane-bound organelles, right? What is a cell membrane? A cell membrane, also referred to as a plasma membrane, selects what can go in and out. So it's called, it's referred to as being selectively permeable. It selects what it permits inside. So for example, oxygen could just go into our cell. It doesn't need any channel proteins. It doesn't need any help. It could just flow in. Nutrients and waste can flow out. And why are they so important? So, it's important because, the membrane-bound organelles compartmentalize different areas of the cell. Like we mentioned, like you can have different rooms with different pHs, different ion concentrations. And so basically it's allowing only certain things in and out of the cell. It's a selective barrier. And so it allows for specialization of different jobs to occur in that little area, right? So, for example, you can concentrate enzymes and substrates in the same physical area. Organelle membranes are usually folded to increase surface area. So some folds are called cystae in the mitochondria, thylakoids in the chloroplast. And so this increases the surface area. And so because there's more surface area, it means that there's more space for important enzymes and reactions to occur there. All right. So it gives them more real estate, so more space for things to work without making the organelles too big. And then when we do our lab, we'll talk about why it becoming really, really big is inefficient for a cell. And so more specialization equals more complex life. So you have more things that do its special jobs. All right. And I challenge you to do these questions so you you check for understanding. All right. All right. So let's talk about the comparison of cell sizes. But before we get into that, I kind of want to explain the surface area a little more. So it's like a difference between a flat sheet and a crumpled piece of paper. So you crumble a paper, right? You crumble it up. You have those little ridges. And so that has more surface area, even though it takes up the same volume. So like, for example, in our body, we have a small intestine. And it has really, really tiny, like, hands, little hooks that are called villi and microvilli. And so this also increases the surface area, and it allows for more things to be absorbed through your small intestine. All right. Why are we talking about the size of the cells? Remember we said prokaryotes are super, super tiny compared to eukaryotic cells. And so this leads to something called the endosymbiotic theory. And so this is a fascinating theory that basically describes how eukaryotic cells evolved. And if you think about it, do we have eukaryotic cells or do we have prokaryotic cells? Do we have a nucleus? And we have a nucleus, so we have eukaryotic cells. And so basically, you can picture like that cells became very complex because we had prokaryotic cells swimming around like 2 billion years ago, and the atmosphere was becoming very oxygen rich, right? And so it used to be toxic to many early life forms, but the large cell engulfed smaller prokaryotes. Instead of digesting them, they formed a partnership with them. And so the small prokaryotes that were good at using oxygen for energy became the mitochondria. All right, so you can picture the mitochondria, which people refer to as the powerhouse of the cell, became efficient at converting things into energy, and it did so by using oxygen. And so later on, some cells also engulfed the photosynthetic prokaryotes that became chloroplasts. All right, so you can see this is the mitochondria, and the chloroplast is right here. I know this is the chloroplast because chloroplasts are green, right? And so this eventually became a partnership. And so eukaryotic cells evolved after long ancestral prokaryotes ingested the mitochondria and chloroplasts. So after a lot of ingesting and a lot of relationships being formed, it formed a close mutual relationship with them. And so the ancestral prokaryote became able to survive in the oxygen-rich atmosphere. Right, we said before oxygen used to be toxic, and so now this oxygen was being used to create energy. So proto-prokaryotes got resources and protection from harsh conditions of early Earth's atmosphere. All right, and so mitochondria and chloroplasts have their own DNA, which is circular, like bacteria. They have their own ribosomes that are the same size as bacteria ribosomes, and they reproduce by dividing like bacteria. All right. All right, and then you have a think about it question. A biologist was looking under a microscope and found a microorganism made up of a single cell. Is this enough for the biologist to determine if the organism is a prokaryotic or eukaryotic cell? If not, what else should the biologist look for? All right, so this is your first question. Your second question is, if you were looking at DNA inside of a cell, what are 2 things about the DNA you could look for to support your claim that the cell is eukaryotic? Again, I also want you to try to do these challenge questions, and then you have 2 homeworks, the AP daily video 2.11 and also a crash course on the tour of the cell.